So I want to talk today a little bit about science in a general sense and biology in an even more specific sense, because I think a lot of people don't really understand fully, unless you have been engaged in science, how the process of science actually ends up working. And even if you've been engaged in some forms of science, they differ pretty dramatically from each other. So I'm going to talk today about science in general, how it back relates to life, the world, and a current virus pandemic, as well as why biology tends to be just a little bit different and oftentimes more complicated than some of the other sciences that you may or may not have encountered. So, basic process of science, most of you know. Most people are familiar with the scientific method. We can take a look at the scientific method in the format that you would be most familiar with. So, most of the time, science begins with some kinds of observation. You got to see what happens. You form a hypothesis, which if you've seen any of my other scientific method stuff, you'll know that I like to talk about as an educated prediction instead of a guess, because scientists don't just take random guesses, they use the information in their possession to know how things work. After that, you need to run some kind of good experiment, and every experiment's a little different. It will provide you with different information. You have to collect and analyze your data, and then you have to put that data out into the world. So draw some kind of conclusions and publication of some sort to get it out there so people can see what you did, look at it themselves, and have an idea of it. This top part is not usually too hard to understand, but we can get a little bit lost in this analyze data section, and I feel like that is sometimes very problematic. So, for instance, data analysis. What does it mean? How do we interpret data? What kinds of things are we actually looking at? Okay, there's a bunch of different things you'll find in data analysis. There are a couple of different ways to do things. We can directly compare different groups. Usually that involves having a study in which one group takes something, one group takes something else. We can look for correlation, which means that we look to see if an increase in one area leads to an increase in another, or the reverse, an increase leads to a decrease. We can look at what we like to call multivariate statistics, meaning that there are a ton of variables and we're just trying to find any pattern at all. All of these take a fair amount of math. Now, math is a little bit more precise than biology. So this is what I was kind of referring to earlier, in that math, and in fact physics and even chemistry, tend to rely very strongly on various kinds of equations. And equations are an important component of all of those things. This thing plus this thing equals that thing, or if you make some kind of mathematical conglomeration, it's definitely going to be equivalent to this or definitely not equivalent to that. Biology doesn't live in that world. Our equations are a little all over the place. They contain so many variables that sometimes we don't know what we're dealing with, and they're often significantly unbalanced. So when you are trying to take something that is equation-based and relate it to something that is not equation-based, it can be complicated. So here are some of the things you're going to run across. For instance, in all of these, you're going to encounter what people call p-values. I'm a huge fan of p-values. I think they are indeed an important statistical tool. They are a probability factor, which means, did the data come out by chance, or is it far enough different from what you'd expect from chance that there must be something else going on? Not always a description of what's going on, but something else going on. So this is most commonly used in the sort of directly compare type areas, where you say, well, were these two groups statistically the same, or was one of them just different enough that it has a p-value? That's where you get p-values. They're supposed to be less than 0.05. So if it's a percentage, that means less than 5% chance of it coming from chance. Most people prefer less than 0.01, less than a 1% chance of it occurring by chance. I like all of those zeros. Correlations do the same thing. We say, is the fact that this one increases compared to that one significant using a p-value, or is it more likely to be by chance, meaning the data is more spread out and any relationship is just sort of, because sometimes relationships happen. So that's why p-values are important. Now, let's talk about this in terms of, though, the word we like to associate with it, which is significance. Significance has one of those words that has different meanings depending on who you're talking to. If you're talking to a statistician, statistical significance is a p-value less than 0.05 or less than 5% likelihood that this occurred by chance. That is considered to be significant. When you are a biologist, though, you need to consider multiple levels of significance. So significant can mean statistically there's clearly a differentiator between these two groups. But does it have a biological meaning? Does it have meaning in a biological system? Does it actually make a change that is noticeable or worthwhile, or in some cases, really important? So, for instance, you can see people kind of play with the numbers on this. There's a chance of cancer increase, for instance, where if you there's a study, there are a lot of other studies that have actually kind of backed off of this, but one of my favorite studies said that your chances of getting cancer, and I believe it was like a colorectal cancer, increased 20% if you ate red meat. And when you actually dove in and looked at the numbers, you had originally like a 0.5% chance of getting colorectal cancer in general. That's general population. And if you eat a lot of red meat, you now have a 0.6% chance of getting colorectal cancer. That makes significance seem a lot different, doesn't it? So these might have the same p-value of less than 0.01 even. But is that a lot of change from a biology standpoint? Probably not. And certainly isn't enough change to make me change my dietary habits. 20% sounds like a lot, but knowing that the actual numbers are much smaller is kind of a key thing. And that goes along with the idea that biology has so many different variables. We simply can't, most of the time, tear apart the different variables that are involved. There are so many things that affect my life and my health that trying to put them in a box of any kind and saying any single one of them is the component, problematic. That's why if you ask any good biologist, so is it nature or is it nurture? Is it, genetics or is it environment? They will all look at you and go, yes. You're like, what? It's like, yeah, no, everything plays a role. Every single thing in some way or another plays a role. What's the size of that role? We don't always know. What's the point of that role? We don't always know. But there's a role, and it might be a statistically significant role. Is it a biologically significant role? We don't know. All right, so let's relate this to what's happening now with COVID-19. Let's be clear, this is a biological problem. This is a virus, which, although not technically a living thing, lives in living things and uses living things. So we're dealing with living things. And there's a lot of components to it. There are a lot of moving parts to this whole thing, just like there is in any piece of biology. And to be clear, nothing short of a very specific vaccine to this very specific thing is going to be the be-all, end-all answer to it. And even that is probably not going to be as much of an answer as we'd like it to be. But that's part of the problem. So every time you hear something, it's always a piece. Pieces are important. So for instance, mask wearing is a piece. It definitively cuts back on a certain number of particles, of which a percentage might be virus containing. Is that percentage statistically significant based on modeling? Yes, definitely. Is that the percentage that is going to specifically keep you from getting or not getting the virus? I don't know. Your life has too many variables. I can't tell you that. And we do know that when we put the large scale data together, that masks are great. But in fact, they only are great when they are also used along with distancing measures, washing your hands, using sanitizer, and keeping track of all the other things we've been told. People's brains don't like that. Your brain doesn't want to just have like 1,000 variables in it. It's way too complicated. We want to nail one thing and be like, this is the one thing. Nothing is the one thing, I'm sorry. But you should do everything available to you to try to gather it together, make those pieces. Drugs are going to be the same way with COVID-19. We are going to have many different drugs, all of which are going to be helpful in some manner or another, at least partially. So for instance, a new article came out today. Coronavirus dexamethasone proves first life-saving drug. Now, first of all, that's not an accurate headline. We have been using other things that we think have saved lives. It's just a matter of whether those things have worked or percentages. So same problem. So item 1, poor headline. Sorry, BBC, you should work on that. It notes that it's cheap and widely available. I mean, that's good. And then it starts to talk about the world's biggest trial testing existing treatments, which is true, not just this treatment, but other ones. And then it talks about some of the things that can be used. Now, from a general standpoint, dexamethasone is a great choice. It is a steroid. It kicks back inflammation, which I've talked about is a huge problem in COVID. And so if we go to the actual site for the actual information, as you can see, randomized evaluation of COVID-19 therapy. This is a group that is working on it. They are doing what seems to be relatively good studies. They have been recruiting and getting things going. And they have been able to publish about this. So now let's talk about significance. So go to their results page, because here's their dexamethasone statement. And their results are right here that when they used it for people who were in the hospital and on ventilators, they did better. So here's our, there we go, reduced deaths by 1/3 in ventilated patients and by 1/5 in other patients. And these have very nice p-values. This one is 0.0003. This one is 0.0021. So what does it mean to reduce deaths by 1/3 in ventilated patients? Do you know how many people who are ventilated actually die? I hate to tell you this, it's most of them. By the time it gets to that point, this virus has already caused so much damage that it is hard to get people off ventilators. Anybody who goes on and comes off a ventilator is extremely lucky. So let's read one more line down. Based on this results, 1 death would be prevented by treatment of around 8 ventilated patients, or around 25 patients requiring oxygen alone. Dexamethasone saves 1 out of 8 people on ventilators and 1 out of 25 people on oxygen. So does it help? Sure, it saves that one person. Should we look into using it? Sure. If it doesn't have any other side effects so far and saves that one person, we absolutely should. Is it the drug that is going to save us? No. No, it is, again, as I pointed out, not the drug that is going to save us, because there probably isn't a drug that's going to save us. So that's important to know. And by the way, that research, although it is data and it has analysis, we haven't seen it yet. That data went actually out as a press release to the BBC before continuing the process of the scientific method, where it would be published and reviewed by other scientists so we can get a sense of what's going on. There's been a lot of studies that have come out very fast and very recently, and some of which the data has been suspect on, and people are pulling them. Be careful. Ask your fellow scientists before you believe something. All right, so knowing all this nonsense about biology, life, and COVID, where does this take you? Well, first of all, now you know that not any single thing is going to be the be-all and end-all to help you, and not everything can save you, but the components do all work together. Everything adds up. Summation is a concept, and that every little thing you can do does help. Don't assume anything is going to be solved instantly. This is going to be a long, annoying process. Get used to it. Be aware that every aspect of risk is indeed an estimate. Everything is estimating, everything is a model, everything is somewhere. We're doing our best to base stuff on that, and the people who are most liable for human life are taking the farthest point. They're trying to be the most careful. You can take your own risk at a particular level, but don't assume everything is a lie, but also don't assume everything is out to kill you. That's the way I usually look at it. But hopefully now you understand a little bit better how science works, how biology might function, and why you should always be careful every time you read any study, even ones with great p-values. Thanks.